Welcome to the IOMP webinar. My name is Mahesh. I'm the Science Committee Chair in the IOMP. I'm also a professor of radiology and cardiology at the Johns Hopkins University in Baltimore, USA. It gives me great pleasure today to welcome all of you. I have with me two great speakers here, and I also want to welcome our IOMP President, John Damilakis. He has joined, and want to thank Dr. Magdalena Stova, who is doing all the technical aspect of this webinar. She is our Secretary General for the IOMP. So this is our final seminar for this 2022, and we wanted to do this in a unique way, and we wanted to do it to early career researchers. And it has come across highly recommended speakers. These two are, in fact, both of them came recommended through my colleagues in the Science Committee. So I would like to thank you for joining. A couple of home stuff, logistics. The question and answer will be answered at the end. So please submit your question in the QA box, not in the chat, so that I will collect it. And after the two speakers finish their talk, I will moderate these questions between the two of speakers. Each speaker will speak for 20 minutes, and then we're going to have a session for 10 or 12 minutes. And it's also great to see so many audience across the globe joining us. And since this is an international crowd, maintaining the time point is really difficult. I know some of you may be up at 3 o'clock in the morning. Some of you may be up still late in the night, 11 o'clock. Either way, we really appreciate joining this webinar. Having said that, I don't want to take up too much time. Let me introduce the first speaker. It is my pleasure to introduce Dr. Andrea Abril. She is an assistant professor and an academic coordinator for the Master of Medical Physics at Bogotá, Pontificia Universidad Javeriana. Pardon my pronunciation, but it's in Bogotá-Colombia. And since 2022, she is an assistant professor at the National University of Colombia. She has taught subjects such as advanced radiation physics, radiobiology, and numerical methods for medical physicists, which has enabled her to integrate experience in Monte Carlo simulation with the radiobiological concept. As I said earlier, these are early career researcher, means they're on their way to fame, and they're working pretty heavily on this subject. And the title of her talk is Low Dose Simulation and the Perspective in Radiation Protection, how this low dose simulation is going to help. So I want to welcome Dr. Andrea Abril.
Thank you, Mahesh. I want to share my screen.
Please.
Let me see. Oh, I don't find it. Okay. I live like this to have a better control of the presentation. So thank you for the invitation. It is a pleasure to me being here. So I will talk to you about around a research project about low dose perspective on radiation protection. So I divide my talk in 3 sections, and the sections are related with the research questions before about the problem. So why to make an approach from the physics to low doses. Then I will talk how to do it, or at least where we started. And finally, I will talk to you about how the interdisciplinary work is needed to abort or to approach to a final conclusion about low doses. And of course, some summary and results. So I want to start defining what are low doses, what we define as low doses. So we know that the main endpoint from the radiobiological process is the cancer induction. So up to date, we have human epidemiological data observed at acute doses below to 100 milligauss and above. So we have evidence below 100 milligrays, but the data are not reliable enough. So the international organizations or committees are defined that limit us the limit of low doses. So the fact is the response, for example, the stochastic effects related with the dose are well defined at intermediate doses, but at very low doses or even very high doses, the response is not defined yet. The model used up to date is the linear non-threshold model. And this model is challenging constantly because could be or overprotective or underprotective. But for now, for today, the current facts and our starting points are mainly four. In general, the statistical power of the epidemiological analysis around the endpoints produced by low doses are very limited, because there is a strong relation with other agents that can produce the same effects, and it's difficult to have consistent studies that can be a response around low doses. And argue constantly represented is the adaptive response. For now, the international organizations refers that the data themselves are inconsistent, and maybe the mechanisms are not well established for now, and maybe some lesions can convert into mutations too. About the immune response or MESIS2, the evidence was examined by the organizations, and is very difficult to define if there is an effect result of the low doses. And it's difficult to define if the low doses can stimulate or suppress the immune responses. And finally, with that current facts, the linear non-threshold is constantly challenged by the studies and the evidence, but the evidence for now don't provide compelling results to abandon the model. So the international organization encourages us to study and develop studies in vitro to robust the information. And the idea is, why study low doses if the evidence is not enough yet? So the reason is because we are exposed to low doses everywhere, in diagnostic tests, in environmental exposition, in nuclear industry. And the main reason is the radiation exposures associated with the human activity are almost low or very low doses. And there is a population important in this study, and is the radiation workers, specifically the diagnostic radiation workers. And what we are looking for when we are study radiobiological process from the physics. So from the physics, we have control into the interaction radiation matter process. So we can link the process with the physicochemical or chemical stage around the production of oxidative or reactive species. And the diffusion of the species serves us to identify where are the lesions, where the energy is deposited, and but up there. Because the relation with the biology needs a complete overview. So a good tool to integrate the information is GM4, but specifically the applications developed by the collaboration of GM4DNA, and the results related with the deposition of the minimum energy, the tracking or the localization of the deposition, could be explored by the application. So we can study the reactive species overgeneration that has a strong relation with some effects, the DNA damage via strand breaks, and obviously, if we know where the energy is deposited, I can explore how is the damage tracking. The endpoints that we are finding are, the main endpoints are cancer induction is the main endpoint, and heritable effects, but the evidence at low doses present some interesting points. There are the induction of non-cancer disease, for example, circulatory disease, cataract, and there are other like hormesis. So how to do it, or how we started, which is our starting point. So we maybe, the applications or the amazing work for the collaboration GM4DNA. They are developed so many applications around the study of the DNA process with the radiation matter. And some two applications permit us to understand the specific questions. How the reactive species are generated and where generated, that's the chemical applications. I can track the diffusion and localize where the energy is deposited. And the other one is the application related with PDB for DNA. This is a very sophisticated application because you can import the geometry of a specific protein into the environment of simulation, and you can explore how is the energy position localized in the protein and associate with a minimum energy, how is the strand breaks, double strand breaks, and localize the process. In this case, it's very important for us because if anybody is working with GM4 knows that the geometry is the most important part in the simulation. And the geometries related with DNA or the protein are very complicated. So you can import the geometry for the interest proteins and define the parameters. So there are too many applications developed from GM4DNA, but I am specifically interested in which one I can reply into in vitro assays. So how I develop the in vitro assays, I change the dose in the region of low doses, and we can examine if there are cellular death, but at low doses it's difficult to see cellular death over the normal limits, or the mechanisms of the damage. So I am interested in the PDB application. So and what we can get. So Monte Carlo makes us the possibility to get from the physical parameters, dose, energy, particle interaction, robust information, because we can reply so many times, depends of the computer possibilities, and explore how are the mechanistics damage about the DNA. The key to have robust information is the possibility to correlation and validation, and the validation is made with the in vitro assays. So just to mention it, the DNA collaboration is very active, and the last year, as you can see in this plot, there are too many papers connected around the topic, and their results are amazing compared with the experimental size. So for us, it's a very good tool to start into to define the minimal parameters for the site. So what do you want? We want to quantify the lesions that exceed the basal limit, and that's just to mention a number, is around 12%. That is the importance of the in vitro side, because we are experimenting single strand breaks and reparation process every time for different agents, difference to the ionizing radiation. So it's different to discriminate if it's for ionizing radiation or for other normal agents in our lifestyle. So it's very important to define the basal limit. So why the interdisciplinary work is important in our approach? Because I can track the physical interactions. I can define how is the physics into the simulation. I can define how particle is, the better approximation, the dose quantifying, but for me it's difficult to define what is the biological target of importance. So it's commonly in the literature or in the papers define the radioinduction damage relate with the cellular death. But here the response or the evidence and the international agreement suggests that the interaction with low doses is different. Is directly to the mechanisms of cell reparation. So we are interested in the molecules in charge of it. So in this case, is just an example. We have protein XRCC1 in charge of the binding process and the reparation process in the DNA. So the first step is identify the series of molecules in charge of it. So that's our first step, and is a possibility to reply in vitro stage. So we part from the first or simple assumption, and is the correlation into the single strand breaks. Makes a future induced change into the mutations, or not necessarily with the cellular death, but we can track this in vitro. So we import that geometry for that proteins, it's just to mention it, and we expose it the protein for different low doses. We are here in a region of very low doses, and the geometry is defined by a surrounding box around the protein, and the dose is calculated around that box. In this case, the particle or the radiation comes from electrons of 100 keV. And the results is we have an increasing number of single strand breaks, and at more or less increase over the basal limit, but the better result is we can find single strand breaks, but at that limit we don't find double strand break. That suggests how will be the size or how will be the methodology for the in vitro size. In this case, I told you about in all the talk. Our focus is not cellular death, is the reparation mechanisms and are strongly correlate with the single strand breaks. Just for summarize, I will have to say that the Monte Carlo simulations provide a good approach for the study of low dose radio-induced effects. But if I want to have robust information, the validation is very important, and the experiments into Monte Carlo simulation permit us to have the specific parameters into the in vitro assay. The correlation or the actual models for the risk in radioprotections are strongly related with the disease or some endpoints, but there is a very long way. So we can relate the reproduction of reactive species or some simple damage into the repair mechanisms, but the correlation goes very long away, and include many factors in the review of the damage. Is very important to analyze over other agents' effects. The implications in radiation protections, I said it, is linked to the effects, but the important fact is an incomplete understanding of the mechanisms of radiation action at low doses is the main or the principal contributor to the current uncertain of low dose risk estimate. So we need to define how is the effect of low doses to define a specific model at this region. Finally, in this work, we are interested in the effects of the exposure of nuclear imaging workers, specifically in nuclear medicine. And the experiments are developed with technetium 99. And the physical conditions in the simulation implies so many changes, for example, in the geometry and in the media of interaction before to reach to the protein. So that's all for today. Thank you for your attention.
Thank you very much, Dr. Andrea. For all those who are attending, I just want to let you know, we will have a question and answer session at the end. Please keep entering the questions, and we'll answer all the questions at the end of both this presentation. For one of the attendees had asked the question, will they get credit? Yes, it's in the information telling all those who have registered for this webinar and attend the entirety, they will be automatically get the CME or CPD credit point will be given. So without further delay, can you stop sharing the screen, Andrea? I want to welcome the second speaker. This is Dr.Sc Nolan Espelon. He is a PhD candidate at the University of Victoria. Again, he highly recommended by Dr. Magdalena. She is also one of the science committee member. And he, as a member of the University of Victoria Excite Lab, Nolan's active research interest pertains to development of enabling technologies supporting delivery of novel preclinical modalities, including spatially fractionated and ultra-high dose, also called as flash radiotherapy. The title of his talk is The State of Ultra-High Dose Rate Radiotherapy, and he's going to talk about the rapid developments of a rapid theory. Dr. Nolan Espelon is studies works in Canada, so the time frame would have been like 3 o'clock early morning, but he chose to fly to Europe, so he's in the middle of the afternoon. He's going to give a talk. So welcome, Nolan, and floor is yours. Again, please enter the questions, and we'll track the questions. And after his presentation, we'll have discuss all the questions. Thank you.
Great. Well, thank you very much. It's a pleasure to be here. And yeah, the title of the talk is State of the Ultra-High Dose Rate, which roughly translates to a general overview of the who, what, when, why, where, how of flash in the modern context. So I'll do my best. It's a pretty ambitious scope of the talk. So without further ado, I will get on with it. Nothing to disclose. So just a brief outline. I want to talk first flash in general, brief intro for those who aren't familiar potentially, as well as the historical perspectives that led to the motivation of it developing in the recent decade. I want to move on then to flash mechanisms as brief as possible. I do go into some detail, but excuse me if some of the slides become bullet points. It sounds like the talk's recorded, so I encourage you to go back and look at some of the details there in just in the interest of time, but hopefully I can cover all of that because there's much and more I'd like to cover. Then I want to move on to some of the beam parameters and time structure for flash and hypofractionation and how that's playing a role, and then also UHD sources for flash radiobiology research and beyond towards clinical translation. So without further ado, flash in a nutshell, what is it? Well, it's basically a novel therapy that characterized by the use of ultra-high dose rates to facilitate the delivery of treatment doses in ultra-fast sub-second intervals, generally as a single fraction or in a small number of hypofractionated segments. The reason why it's getting so much attention derives from the fact that there's a repeated observation of reduced normal tissue toxicity, i.e. this flash effect, without compromising tumor control in a number of animal models, and therefore affording an increased or widened therapeutic window. It's been demonstrated in a number of animals, including veterinary trials in mini pigs, cats, and mice at a number of endpoints. And there was even a first in-human trial with superficial electron treatment in 2019. What FLASH is not, however, is a cure-all or necessarily a replacement for optimized conventional radiotherapy regimens today, at least not anytime soon. But it may help to widen the therapeutic window as in the context of being a boost. And there's a lot more that needs to be done to solidify what the modifying factor is and in what context it's appropriate to use it. Part and parcel to that is the fact that optimal delivery parameters and mechanisms are still largely unknown, but a lot of smart people are working really hard to figure that out. And that late effects still require further investigation, bearing relevance to some of those classic pictures that I showed on the last slide. In a follow-up long-term toxicity study for a veterinary phase 3 trial with cats and mini pigs, they actually did not see the same FLASH tissue sparing effect that they did in the dose escalation trials earlier, and that was attributed to maybe an increase in biologically effective dose, but importantly, differences in beam parameters, notably longer radiation time and larger field sizes. But good tumor control was still maintained. So that does bear importance for the future of FLASH, just that it points towards caution that needs to be taken towards preclinical translation or translation of preclinical research, Speaking of translation, though, clinical trials are underway, including the FASTA-1 trial with scanned protons, which looked at extremity bone metastases, eliciting the comparable pain relief as to conventionally dose rate delivered X-rays. So that's bringing you up to date. But now I want to bring you back to the beginning. Why are people looking at ultra high dose rates? And it kind of derives from some studies in the 1960s, whereby an increased cell survival fraction was found due to radioresistance conferred by transiently induced hypoxia in target cells that was attributed to this radiochemical oxygen depletion hypothesis. Now, what did that manifest as? Well, it came across as this hockey stick shaped survival curve in vitro, which demonstrated an inflection towards an anoxic type response away from the oxygenated response at a breakpoint or a threshold dose that depended on the initial partial pressure of oxygen. Following up to that, there was a split dose study that well demonstrated when you allow enough time for reoxygenation that you actually kind of work backwards and you reduce the cell survival fraction, and you in fact return more towards that oxygenation, that full oxygenation curve, which really points to a role for oxygen. But this is where I want to stop and emphasize that in vitro oxygen depletion with UHDR is not FLASH in the modern context, but it does highlight that oxygen plays a role and that ultra high dose rates can modulate biochemical, radiochemical cascades that might be relevant towards cellular function and response. So with that, let's look at FLASH in the modern context with a big, bold disclaimer saying this is meant to be an educational exploration, not an authoritative explanation. I'm not an expert on the radiochemistry and much of the biology, but I try to be a student of it also. Hopefully, I do it justice here. Starting off, I want to kind of harken back to that original oxygen depletion hypothesis, which put simply is just that application of ultra-high dose rates leads to rapid radiochemical utilization of oxygen, leading to transient hypoxia, leading to improved cell survival, which should, in theory, be correlated with normal tissue sparing. Well, why might that be the case? And the idea is basically that primary water radiolysis products, due to the interaction of radiation in water, are efficiently scavenged by the oxygen, which effectively depletes the oxygen on short timescales before reoxygenation kind of competes with that. And with a big asterisk there, it's technically faster than the free radical recombination rate. This will come back a little bit later. What's the big deal here? Well, by depleting oxygen, you aren't able to fix the damage on important biomolecular targets. For example, you might have a DNA damage chain without addition of oxygen. It's not going to turn into a peroxyl radical that's then chemically irreparable. And instead, on long enough timescales, the damage, the indirect damage, for example, can be repaired chemically. And also with this modulation of oxygen concentration, you might have a lower cytotoxic reactive oxygen species or ROS yield, which bears importance to the survival of cells as well. So what does this kind of bear out to? Like, what is the deal with depleting oxygen, and how does that basically manifest as a flash effect or a protective effect? Well, one model did a really good job of showing how the presence of oxygen or reduction of oxygen towards regions of relative hypoxia reduces the probability of interaction with an oxygen molecule and, say, a damaged DNA, which therefore reduces the probability of that DNA damage being fixed and chemically irreparable, which I kind of alluded to. How this manifests is effectively a shift in the OER, which is basically a manifestation of this oxygen effect, or this based on this oxygen fixation hypothesis that would manifest in a shift in the OER curve that for mildly hypoxic tissues or cells elicits a very large flash effect or difference in this oxygen enhancement ratio, i.e. it's a dose modifying factor modification. And but if you're too oxygenated, if you have too high of a partial pressure of oxygen, that effect would be largely diminished. But also the argument goes the same way for if you're in anoxic or severely hypoxic scenarios, there isn't as much room to have a protective effect. Now that's kind of one of the things that's invoked for why there might be a differential effect between normal tissue and hypoxic tumor tissue. But there's a few holes in this. So this works as long as the partial pressure of oxygen is not too high. Unfortunately, physoxia is generally considered to be between 20 and 50 millimeters of mercury. And depletion rates are such that in order to deplete O2 at these levels in a short enough time scale, you would need almost certainly toxic doses. And zooming out, this figure is really good at showing you for a range of tissues why this might be a case across the board and how there's not really anywhere to hide for this kind of TOD hypothesis. That being said, all this points to is the fact that O2 depletion, like complete depletion, does not appear to be the primary factor. It doesn't mean it's not a contributing factor. But I do want to also see if there's anywhere we might be able to reconcile. Is there any way we can salvage this theory? Because the oxygen seems to be important and a lot of people are on the side of oxygen. And that's where we can also get into some interesting territory of invoking the possibility of stem cell sparing, the idea that stem cells may live in hypoxic niches at some distance away from the source of oxygen, i.e. the capillary network. And based on diffusion models and the physical basis of oxygen being able to replenish over time, so reoxygenation diffusion kinetics at certain distances from tissue, it is possible that there could be a flash oxygen depletion mediated radioprotection at distances from the source of oxygen where stem cells may live. This was further corroborated in a more recent paper by Rothwell et al., who actually highlighted that for such diffusion-limited hypoxia models, however, there's important sensitivities to biological and radiochemical aspects as well, which currently have really large uncertainties, including, and they invoked also uniquely the idea that there's porosity. There's this idea that tissue is not homogeneous pure water, and so diffusion can be very different, as well as the importance of metabolic consumption of oxygen in the cells, and that can really modulate basically how much of a depletion we need in order to elicit a clinically manifest effect. So I'll come back to that briefly, but I want to first put a nail in the coffin. These models really depend on this radiolytic consumption as one of the key parameters. And unfortunately, time and time again, it seems that we show that there's an insufficient or rather low degree of depletion of oxygen at ultra-high dose rates, and in fact, lower at ultra-high dose rates as opposed to conventional treatments due to, if you remember the asterisk, free radical combination, which in effect in one paper by Janssen et al. In 2021, they postulated reduced the steady state concentration of the solvated electron, which in turn means that's not available for oxygen to scavenge and therefore results in a reduced oxygen depletion rate. This is important to feed back into these models. And there's a caveat, though, that such measurements need to be done on sufficiently short timescales because rapid recovery of oxygen, you need millisecond time resolution. Now, while that may not have been the case in the Janssen et al. Study, and I'll quickly go through this slide, don't worry, it has been done in other studies whereby phosphorescence quenching oximetry measurements at up to 3 kilohertz have shown in effect that, yes, there is these depletion rates are kind of consistent, and also there is reduced depletion at ultra-high dose rates as compared to conventional dose rates. And that bears relevance to these models. But perhaps even more surprisingly is that there was a lower G value in tumor tissue as compared to normal tissue. And this correlates with the depletion value that depends on oxygen concentration. That too should be fed back into those models. But it should be noted that not only have these measurements been done in vitro, but also in vivo, but only in the extracellular space. When we talk about this depletion and the biologically relevant downstream reactions of oxygen depletion, it happens in the intracellular environment. And we also know from these models that it's quite a dynamic situation, and there's quite rapid oxygen gradient that is set up. So the fact that oxygen outside of the cells as opposed to inside of the cells might be different isn't so crazy in my mind. It's something worth looking into. But all I'm doing is poking holes in this theory. I want to reiterate that there is a support for the role of oxygen. If it's not oxygen depletion, what is it? Not sure. But again, although it's not necessarily flash, there is consistently a demonstration of a sensitivity of cell survival on oxygen, but there's also in vivo evidence that when you provide hypoxic conditions, you start moving towards a flash-like protection of response. And if you improve perfusion or improve oxygen availability, you actually abrogate or remove the flash effect. So again, probably can't reconcile flash protective effect with radiochemical TOD alone. We can partially invoke the stem cell sparing and hypoxic niches and maybe an underestimation of an OER shift based on the radiolytic consumption and a porous tissue model. But even if we can't do it alone, it doesn't mean it's not important. And that's what moves me on to one last hole poking slide. I apologize, but this is all talking about normal tissue sparing. We can't forget that there's also an important distinction here that flash also creates a differential effect between the normal tissue and the tumor tissue. So how can we get isoeffective tumor control if we're doing this, having this sparing effect that presumably would be somewhat equivalent between tumors and normal tissue? Well, the argument is that, well, tumors are hypoxic. That's been shown to not necessarily been true. They can have a heterogeneous hypoxic status at different locations. They can be well perfused, poorly perfused, intermittently hypoxic, acutely, severely, mild. And so the variation, the heterogeneity in there kind of makes this a bit more complicated than just saying a tumor is hypoxic and therefore you can use the TOD to invoke the differential effect. In addition, what about sparing in well-oxygenated tissue or aerated cultures? That's not necessarily easy to explain when you have an abundance of oxygen and rapid replenishment of that oxygen. So, and there's also the one thing we haven't talked about yet, which is the formation and rate of production and consumption of reactive oxygen species, as well as differences in redox biology between these different tissue types. So unfortunately, in the interest of time, I'm going to move relatively quickly through that, but I do want to touch on one aspect of the reduced ROS cytotoxicity side of things, and that is, well, in earlier kind of papers, there was this postulation, such as by Spitz et al., that there might be actually a larger oxygen depletion due to biologically relevant redox-activated Fenton reactions. So you have the superoxide that's going and activating the release of iron, lab iron, that can then go and react and create more hydroxyl ions that can go do more damage. And then also to these lipid peroxidation chain reactions, one of which I've actually shown below. That basically generates, not only does it consumes oxygen, but it more importantly produces these cytotoxic hydroxyl or peroxyl radicals that can do a lot of damage if they accumulate in the cell. So if this is happening, and this is happening in all cells, maybe it's a differential effect in the healthy cells with improved antioxidant activity that could account for a differential effect. So that's one aspect. One thing too that kind of helps with the idea that, you know, oxygen depletion is too low or too slow is actually was well described in a paper by Labarbe et al., which proposed that there was a radical recombination as the driving factor. So using this kind of brief reaction down here, if you had an indirect damage to organic species producing these radicals, that could then be fixed, the damage could be fixed by the agent producing the peroxyl radical, which when in the presence of unsaturated fatty acids could undergo this lipid peroxidation chain reaction to produce more of them and consume more oxygen. The rates of radical recombination are actually high enough at ultra high dose rates such that there is more radical recombination and less production of tissue damaging peroxyl species. So that was one of the ways that this could kind of be reconciled. And interestingly, there was actually recent evidence to support that lipid peroxidation or evidence for lipid peroxidation was decreased after flash with MT X-rays, which I've not shown, but that was an interesting kind of corroboration of that fact. So again, differential effect, maybe it's differences in cellular redox metabolism. That could be, you know, again, we can account for the flash effect in normal tissue, but if we're not explaining the ISO effect, we need to do that as well. So this was one of those aspects, but there's another one that I want to highlight because of a topic I want to touch on the next slide, which is that tumor tissue radioresistance from, say, TOD or, you know, adaptation to oxidative stress could be compensated for by impaired DNA repair mechanisms if the tumors are hypoxic. And one interesting thing that has kind of borne out in the literature is that indeed there is a subset of DNA damage, in this case indicated by this 53BP1 foci, that was reduced in aerated lung cells and less persistent following flash as opposed to conventional, and only in normal cells. So that's important for not only the fact that we have a preferential sparing from flash, it's differential in that it's only happening in the normal cells, and this reduction in DNA damage is not happening in the tumor cells. Furthermore, as it relates to late toxicity, there's been indications of down-regulation of indicators for normal lung damage, as well as a reduced number of senescent cells and markers for senescence. What does that mean? It means that flash spares progenitor cells from replication exhaustion, and senescence itself has been associated with long-term radiation toxicities, such as lung fibrosis, which has traditionally been seen to be reduced after flash in some of the earlier flash studies for mouse lung, for example. Moreover, epidermal stem cell sparing has been noticed along with regulating repair pathways, flash protons in the irradiation of skin. And so all of this really points to, again, there's the sparing effect, perhaps a preferential sparing effect for the stem cell compartment, which, if you remember back, might be consistent with this physical model of maybe where the sparing, where the oxygen effect could be most important is at some distance away, where in these stem cell niches or in compartments where they're more hypoxic just on average. Even if you're measuring something in the extracellular space on average, that's a higher oxygen partial pressure after irradiation, there might be these hypoxic regions that are more preferentially affected by flash. So moving very quickly now moving into the immunological side of things, inflammation is generally not seen as much after flash. In fact, there's a reduced upregulation of pro-inflammatory, pro-apoptotic cytokines, notably TNF-alpha, but also TGF Beta, which I want to highlight specifically because that has been found. I apologize for the text, but again, hopefully you can go back and read the slide if you want. It's associated with reduced immunosuppression. And in fact, there has been observation that there was improved recruitment of T lymphocytes into the tumor when treated with flash as compared to conventional RT. So whether that's one-to-one with a TGF beta reduced upregulation following radiotherapy or a cocktail of other factors is uncertain, but it's just interesting to see that there are these directions in which these various factors go such that it supports both immune response and preferential sparing based on modulation of the DNA damage response and such and so forth. So that is one more slide on the immunological response. Is probably the most simple one, is that if you are irradiating using ultra short durations, that you provide less time for circulating immune cells to be irradiated or to enter the field and be damaged. So this is just to say that an obvious immunological effect could just be that for non-TBI flash, you're irradiating far fewer immune cells and keeping a lot more that are viable, which could improve immune and adaptive response later on. For TBI, however, it's interesting to note that there's also been a subset of blood stem cells whose functionality has been preserved at relatively low dose for flash radiotherapy as well. So even for a full body irradiation, again, these blood stem cells, these hematopoietic stem cells have been preferentially spared in mice that have received these after irradiation. So that's quite interesting. But I do want to highlight here that the immunological component can only be partial mechanism because a flash effect has been observed in immunocompromised mice as well. But it's just interesting to highlight that there's all these things that could be contributing. And in fact, it may not be one or another. It's probably a combination of all of them in some configuration. It's really interesting to be able to learn more about the radiobiology using flash as a probe in that way. Now, moving on to the second segment, which I will go through much faster, which is how to flash. How are people doing it nowadays? So first and foremost, the importance of delivery on a large temporal scale, hypofractionation, which is important, very important clinically. It's used to achieve higher biologically effective dose. And there was a study by Montegru et al. In 2021 that was able to demonstrate isoeffective tumor growth control with improved cognition sparing for hypofractionated regimens with doses that were dose per fraction that were not too high, but also not too low. So the low dose per fraction not doing anything may be consistent with this idea of a dose rate or a dose threshold for flash, but the ceiling to a dose per fraction is also quite interesting and important for preclinical translation type experiments. Moving on to the microtemporal aspect, and that's just the fact that beam parameters for flash are varied. There's the pulse duration, the pulse repetition frequency, average dose rate, instantaneous dose rate, and depending on the source you're using, whether it's sync or cyclotron, LINAC, superconducting LINAC, the pulse structure is going to be different. So what matters and what parameters, delivery parameters, do we need to be focusing on to make sure that we can elicit a flash effect reproducibly? At the very least, there's been consistent evidence to support that it's the average dose rate, or rather time of irradiation, being short enough that can promote a flash effect, as opposed to it a dose rate in the pulse or an instantaneous dose rate being a driving factor. But that more should be done on that. In fact, one brave group more recently did do just that. They used 6 MV electrons to basically show that there was a sparing in the intestinal crypt cells were correlated with increasing average dose rates, so improved sparing with increasing average dose rate, but that temporal parameters did apparently matter. There was better crypt survival with more pulses, lower dose per pulse, but also a higher frequency, shorter time between pulses. And it's ambiguous as to what aspect of that delivery was the driving parameter there, because at going to slightly higher dose in older mice, the DPP and / pulse number relation inverted. So now it's not quite clear if it was, you know, what thing needs to be changing there, and if it is sensitive in this case just to the fact that these mice at these different time points were more or less radiosensitive. But at the very least, the sparing still increased with increasing average dose rate, even at that higher, more toxic dose level at the more radiosensitive mice older cohort. So that was really interesting, and just skip past that. But also just to note that there was less damage to the gut microbiome for FLASH in that study as well, which was super interesting. Finally, the sources that are available to deliver FLASH for research and beyond. Right now, predominantly electrons, low energy, or X-rays, also low energy, or protons, high energy, which is really good, especially if you want to move towards clinical translation faster. But future technologies looking at improved depth of penetration, VHEE or MV Bremsstrahlung sources for electrons and X-rays respectively, and also using, for example, if high dose per pulse is an important parameter, synchrocyclotrons for pulse proton delivery, as well as delivery of heavier charged particles like carbon ions could be quite important. I'll go through these slides also pretty quickly, but I encourage you to look back at them as reference. Electrons are everywhere. They're ubiquitous in the literature and effective for radiobiology research because they're much easier to get these ultra high dose rates with. We've got the Aureotron prototype system used by the Shoot group and others. We've got the Mobitron IOR type T system, as well as various modified clinical LINACs from various groups around the world now. And it's obvious that superficial treatments are possible with these types of sources, but we need a solution to the depth problem. And that solution will come with very high energy electrons. And there's a few examples of where that might come from. Recently, I learned about the DEFT project looking at X-band accelerating structures up to 100 MeV per meter with multi-beam delivery and fast magnet switching, which could be really interesting. Otherwise, the future phaser concept using similar, I believe, X-band technology. Caveat is that today, VHE flash research is possible and active at CERN. They have the CLEAR facility, which goes up to 200 MeV, and there's some research being done now. Very interesting to look towards how we can drive VHE accelerator development in the future. Moving on to X-rays, can't not say X-rays, they're my favorite. But currently, source of ultra high dose rate X-rays are not so readily available. You can use X-ray tubes at short SST, for example, with fast shutter systems to elicit pulse control, there's a group doing parallel opposed rotating anode sources with higher power that can elicit better field conformity and depth uniformity for small animal research. That's really important. Additionally, for superficial stuff, there's synchrotrons that to a few hundred keV that are suitable for preclinical research and have actually been used effectively by Montecristo et al. Back in 2018 to show sparing of the brain using ultra high dose rates delivered by a synchrotron. And there's also now, I got to toot my horn a little bit, MV sources with superconducting LINACs at 2 facilities that I know of, one at Triumph, which we helped develop, and then also at the Chengdu Free Electron Laser Facility, which have been doing radiobiological experiments for a couple of years now. The problem being that they're short SSD and not really portable infrastructure, and they're superconducting. The goal then is to allow for improved penetration at higher energies using existing accelerator technologies, and so that we can use IMRT and adapted planning systems, which are well understood. But the problem is that there's low conversion efficiency when you use a Bremsstrahlung target, and that's difficult to manage. So novel solutions have started to come up, including this phaser system with the Sphinx collimator that combines intensity modulation as well as using E-beam raster scanning on the long collimator, as well as a Bremsstrahlung conversion kind of all in one. And then there's also this ROAD system by Liu et al., whose goal is to intensity modulate so that biological gain is not offset by the lack of physical dose conformity. I liked that quote. And in the future, maybe laser-driven Compton sources. Right now, really, KeV energies are—
Nolan, you had 2 more minutes.
Okay, sounds good.
A lot of questions.
Yeah, sounds good. And then I can't stop. I have to say protons, so I'll say protons and I'll be done. It's just that those are readily available and UHDR compatible. They reach clinically relevant treatment depths with good conformity and dose fall off. But new systems do have high capital cost, but existing systems can be adapted, as have been done already, and clinical trials are already underway. The issue then is just there's complications with the dose rate definition. Is it the mean integrated dose rate, the in-voxel mean or instantaneous dose rate, or the spot peak dose rate? The effects of variable dose rates and the fact you might not always be reaching a flash dose rate threshold in the course of delivery, effects of that are not unknown, and the aspect of repainting with pulse delivery in PBS is also up for debate. So treatment planning is complicated not only by RBE changes with the higher LET, especially in the SOBP, but also due to the influence of the flash effect that might be variable on a voxel by voxel basis. So that brings the question, is it pencil beam scanning or scattered? There's complications to both. In the scattered beam, for example, it's harder to get sufficient proton beam current for large clinically relevant field sizes. SOBP versus transition mode, it's great to leverage the ballistic properties, but apparently improved sparing is found with either, but improved tumor control with the SOBP. So that's all really interesting. I will skip the high LET considerations, but I will get the punchline, which is that it's also been found that there's normal tissue has been spared with comparable tumor control using carbon ions. So that's really interesting in the light of the different kind of radiochemical and biological implications of using high LET radiation. So with that, I have optimism for a flashy future. I will leave this slide up, and thank you very much for listening to my talk.
Thank you very much. Sorry for rushing you.
Oh, no.
But I could see the excitement in both of the speakers. Each of you can speak for an hour, but I know. However, I also want to let you know we have a lot of questions. And as of now, I'm really happy to say that we had more than 1,122 attendees around the world watching you. So, and I just also want to tell the attendees that whoever registered for the webinar and stayed for the whole hour will get the credits. IOMP will credit. Plus, IOMP has recorded the webinar. It's going to be on the website soon, and you can view any time. The second thing is I also want to tell you, we have an exciting program for the next year. Already we have two webinars signed up. We're going to start the new year on January with Dr. Chris from South Africa, going to talk about safety blinded or safety minded. It's an interesting talk. Don't learn safety by accident, all those things. The second one will be on carbon ion radiotherapy, will be in February 7th. So I would urge all the attendees to participate and provide the same encouragement for the speakers. The few more webinars are already being lined up. It will be announced pretty soon. Having said that, Nolan, can you take off the screen or stop screen?
Absolutely.
So what I want to do is I'm going to share the one question at a time for each of you and try to answer as briefly as possible because we have about 20, 30 questions. I'm not sure how many we can handle, but we will try to get as many as possible. So the very first question to Dr. Andrea. So the question is, as this low dose is defined as less than 100 milligray, how do you define low dose rate? How do you do? Yeah.
Okay, the UN agency puts a limit around 0.1 milligray per hour, okay. But I didn't remember if the ICRP put a limit of 5 milligray per hour.
That's correct. Yeah, 5 milligray per hour. The next question for you itself is, damage to DNA is direct or indirect process. So how do you represent indirect damage parameters for the free radicals formation in your simulation?
Okay, nice question. So the protein is embedded in a water media. And when the radiation hits on the media, you can follow the reactive species. So you can track the position and you can track the energy deposited into the absorption. So you can correlate the minimum energy by the surrounded structure around the protein and make a relation into the indirect damage.
Okay. The next question to Nolan. Again, gave an excellent talk. Time was not enough, I understand, but there's so much to go. So the first question is, has the flash effect on the immune system been investigated?
So to my knowledge, there has been investigations on, I believe there was a paper looking at, I think it was Durante 2018, I hope I got that right. And it was looking at the circulating immune cells and finding effectively reduced chromosomal aberrations in light of that. So there has been some research looking at just preservation of circulating lymphocytes, but I think there's also, like I said, these stem cells, these hematopoietic stem cells that can differentiate and effectively be important components of recontributing to your immune response over time, especially if you irradiate and you lose some. The fact that those might be spared at least somewhat preferentially at lower dose flash is also interesting. And this is consistent with the idea that there's this stem cell sparing capacity of flash, which I find is really interesting, whether it's derived from that physical model I was talking about or some other mechanism. It's super interesting. But there has been research on that front. And there was also, excuse me, I'll just
I think we can go to the next question, which will we can add up. What is the clear-cut difference between flash and clinical Linacs? In less than 2 minutes.
Okay, so the depends what you're talking about. So a clinical Linac is generally modified to be pulse width. It's a high dose per pulse, higher rate. So it's people also just kind of turn the safeties off, as I understand, and put the samples they're willing to irradiate much closer to the and they remove the scattering foil. I think sometimes they can use it in, they can leave it out, and by also reducing the distance, you're increasing the dose rate. So people are generally putting their samples or their animals or whatever in the treatment head effectively to increase the dose rates for modified Linac flash, as I understand it. Otherwise, you just need high enough current, like VHE beams or otherwise low energy electron beams can have very high charge per pulse, even if they're only delivering one charge or one pulse with a very low duty factor, either not. So they're just much more efficient at creating ionization and delivering dose. So if you're not careful, it's probably easier to deliver ultra high dose rates as opposed to not, if you're not going to scatter the beam, for example.
Okay. So there were a couple of questions asking whether they can have the slides. I don't think they can have the slides. That's one thing. This information is available on the webinar, so they can always view and visit back. And people have asked about your email addresses. I think most of you gave your email address at the end of your slides, so they can go back to the webinar and view it. If not, we can provide the email if they want to contact. The next question is for Dr. Andrea. The question is like, do you use some mathematical model, radiobiology, and compare the outcomes? So to compare the outcomes.
It's difficult to employ a mathematical model because there are too much. And someone says, do you have a region of radio sensitivity or other claim a region of radio resistance? But the general principle is the polynomial model. So you can adjust the data, and the responses are very similar to UV exposition, but I don't have one specifically.
The other question in the same line, since you work at low dose with bad statistical correlation, how can you improve the statistics or accuracy of the result without increasing dose? So how much photons you need for a good statistics, but not high dose? I don't know. Yeah.
It's a difficult question because in computation it's simple. You can increase the number of events, and the number of events means different proteins, and the statistics is more related with the dose. So we increase the number of particles and increase the dose, but the statistical result is different. So I have to simulate a collection of more molecules.
So one more question on the same line regarding the low doses. At what level do the predicted effects of low dose radiation exceed the observed natural error rate in DNA daughter? It's a complex question.
Yes, it's very complex because, as I say in the talk, we are exposed to many agents that presents or induce similar effects to low doses, but we have a number in mind, and around 12 or 30% of single strand breaks we can relate with another agent different of the natural exposition to other agents. So we are looking for what is around this limit. So that there are too many components.
Okay, question to Dr. Nolan. How can you achieve beam intensity modulation with flash beams?
Yeah, so that's a really good question. For electrons, it's a tough one, probably a raster scanning sort of methodology. Otherwise, if you had beam shaping and you had multi-directional, simultaneous multi-directional capabilities, then maybe you would preform the field based on a treatment plan or something like that. But electrons, I think are tough. I think scanning is a good one. Proton beams, I think it's already kind of built into the radiation modality. It's there by way of proton pencil beam scanning, pardon me, in particular. But also people have used, I think for scattered beams, you could use a sort of filter that would also preform and produce intensity modulation. X-rays are probably the most interesting because we can't use gantries anymore. We can't use multi-leaf collimators in that sense because the gantry rotation isn't compatible with this kind of sub-second delivery timescale. So you'd be looking at, like, the Sphinx is kind of cool in that it's using intensity modulation by way of irradiating different parts of the target and kind of eliciting a pencil beam, almost a grid-like pencil beam array that it rasters the beam there as well. So again, that's a fast beam control, magnet control that would be really critical for that. And then, yeah, that would probably be where I would start.
Good. So the one more question for you. Were these tumor bearing or normal mice you were doing on this experiment?
Which experiment?
You were talking about some of this mice experiment on the flash.
Yeah,
so they were telling, oh, go ahead.
Yeah, there's been both. There's experiments just looking at normal tissue toxicity in the various organs of interest. You got abdominal radiations, lung radiations, brain irradiations, but there's also tumor bearing animals. I'd say like the tumor control side of things is much less well documented, but there are papers by in particular the Shuv group and now Montagu et al. And his colleagues that have looked at tumor control. So there are tumor bearing mice that in various sites for like sarcoma, for example, and glioblastoma that have been looked at. So
one more question. This attendee appreciate your talk. Like most of the attendee questions we get is prefix telling that an excellent presentation. So I just want to convey for both of you. They are telling like, I'm not familiarized with the flash RT. So I wonder if there are any type of organs or cancer that are currently considered unsuitable for flash treatment.
Yeah, I mean, it's a really great question. I don't think I have the answer to that offhand, but I think that's one of the questions that needs to be answered right now. Like if there are certain, there's papers out there where clearly you see more or less of a flash effect, but to kind of decouple that from, well, is that a beam delivery optimization problem? Is that related to individual sensitivity of the animal, maybe the murine, like the type of mouse you're using as opposed to, and male, female, or if it's specific to the tumor site. I think ideally, one of the areas that you want flash to be good is where you're allowed to kind of dose escalate, where you can leverage this this reduce normal tissue toxicity so that you can deliver higher doses to treat otherwise very difficult to treat tumors. I think central nervous system targets are really, really interesting for that. But as for the reverse, what really you can't treat, I can't really say. I'd say something like if we believe oxygen has a role in the lung, maybe would be a tough one, but that's not the case, obviously. That it does seem to have an effect in the lung. Abdominal radiations, I think, are really, really tough.
One other question. Can flash help explain the Kakia effect seen in most patients undergoing radiation therapy? It's spelled as CACHEXIA.
I'm unfortunately not familiar with that effect.
Okay.
Is anyone able to help me, or is that?
I'm not familiar with, because I'm a diagnostic imaging physicist, so I don't want to venture in that area right now.
Apologies, I don't.
No, it's okay. No apologies needed. So there are a couple more questions. Two questions has come, which is probably I could answer, or I can give it to you and then you can tell. One said, could you please let me know of the side effect of mammogram radiation? If you're not familiar, I'm happy to answer the question. Do you want me to answer, Andrea, or anybody?
So, I didn't remember the specific energy from mammography. I think it's around 100 keV.
Less than that.
Yes, and the dose could be reached to 102 milligray or less.
Yeah.
Yes, there are in the region of low doses, but maybe the increase in exposition could be an effect. So there is another.
With regard to mammography, the general understanding is like the effective dose from mammography, a typical screening mammogram is about less than 0.5 millisievert. And it's considered one of the very safe screening model. That's why it is quietly approved for screening asymptomatic patients. So the radiation in mammography should be much less. The risk is so low compared to what its diagnosis, because some people will scare to get a mammogram and they might end up getting cancer, breast cancer. So that's where the screening mammogram comes very helpful. There's a second question telling or doing variety of security scans at various stations and mall and many more, what's the radiation? Again, for these two things, we have published some papers on this one. I would invite attendees to visit a website called radiologyinfo.org. Radiologyinfo is one word.org. That website is predominantly done for both the patients and the radiologist. It has a lot of this information. It also has a dose table. Regarding the security scan, the security scans these days, there are two types. One is the radio frequency a screening center, which is all in the US centers. They are more like a booth you stand inside, and that's a radio frequency. There is no x-rays. There are other type called the backscatter x-ray, where you stand between the two big refrigerator, and there is a low dose scanner goes and scans and provide a surface image. The dose in those things are also very less. The equivalent is like you need to get about 2,500 scans to get an equivalent dose of one chest x-ray. So I just want to inform that one. Having said that, one more question for, let me see. So for Dr. Andrea, is it possible to use MCNP5 for the same purpose as Genet4 DNA to study the effect of low dose?
I didn't use it before, so I just simulate with GM4. So I'm sorry, I don't know.
No problem. No problem. So the question to Nolan: how did you manage the dose rate measurement in FLASH regarding to pulse nature of the radiation, the metrology and dosimetry and so forth?
Yeah, that's a good one. I unfortunately would have been a 2-hour talk if I talked about the dosimetry as well, but it's tough when you use really high dose per pulse. You run into the issue with when you want real-time management in the very conventionally used ion chambers. You're going to run into the issue of lots of recombination and needing a really, really high correction factor that may come with a lot of uncertainty and such. So I think what is usually safest for pulsed flash radiation with reasonably high dose per pulse is a lot of people are using would have been deemed dose rate independent dosimeters, such as gap chromic type films, thermoluminescent dosimeters. There's been some success with solid state detectors such as the diamond, the micro diamond, or the flash diamond, I think they have now. So I would think alternatives to gas filled ion chambers is a good start. If you have a more quasi-continuous type beam, then it's like in a lot of proton beams, it's a lot more tractable. But there is a limit. There's a definition there that you can find in the literature. Yeah.
So one of the attendees, Dr. Gamal Akbani, answered our question about cachexia, CACHEXIA, and he mentioned that this causes predominantly muscle wasting after radiation therapy due to loss of stem cell population in normal muscle tissue. Thank you, Dr. Gamal. There is also one other information only, but I cannot explain the whole thing. This is from Dr. Ruth McCallum. She's telling they are based in UK. They got a major grant for studying the ion radiotherapy. So probably if you guys, anybody interested can Google LHARA, and in UK, it's a long website, so they can find more information. They're also going to do some workshops next year. So I think we're coming to the end of the discussion. We may have missed one or two questions. Please excuse us. But and I also want to purposely thank all the participants. It's really amazing for the work we do when we have more than 1,000 attendees participating in this webinar. And we are very grateful. I also want to thank the two speakers and Dr. Andrea and also Nolan. And I also wish you best for the future. And for all the attendees, please visit the IOMP website. In a day or two, these recordings will be available. You can view as many times as you want. And finally, I also want to thank our president, John Demilakis, for providing this opportunity, developing this whole webinars. And also Dr. Magdalena Stova. She is the technical expert behind the scene, and it's really nice. And so I want to thank, and also personally, I want to thank all the attendees and all of you. I think we're already over the hour, which is good. And thank you.
Thank you.
Thank you.